Welcome to the debate. For over a century, biology was governed by a very simple, almost brutal rule. If you couldn't grow it in a petri dish, it practically didn't exist. You were essentially invisible to science.
Right. The agar plate it was the ultimate biological gatekeeper. It was a comforting system because it gave us things we could see and touch, but it was incredibly restrictive.
Restrictive is almost an understatement. But then we cracked the genetic code. We started sequencing DNA directly from the environment, and suddenly we realized the petri dish was blinding us to almost the entire microbial universe.
Yeah, which brings us to the foundational divide we are exploring today in modern microbiome science. And this emerges directly from recent comprehensive source material analyzing this exact tension.
Exactly. The core question is this: has the field become excessively dependent on sequencing-based inference at the expense of actually growing things? Like, are we trading experimental microbiology for computational models?
Right. It's this fundamental tension between mapping the genomic shadows of a community and holding living, breathing biology in your hands. And looking at the recent literature, I firmly argue that physical isolation, what we call culturomics, is absolutely necessary because, without the physical organism, we just cannot validate biological truth or prove causality.
And, well, my read of the data is that culture-independent omics is the only way to bypass cultivation bias. I argue that we desperately need sequencing to capture the full breadth of the microbial universe, right? Without destroying its true ecological context.
I understand that perspective, but we have to look at what we're losing in the process.
Well, let's look back at why we even needed this computational revolution in the first place. There is a historical reality in our field called the Great Plate Count Anomaly.
Ah, yes, the absolute bane of classical microbiologists everywhere.
Exactly. For decades, researchers would take a scoop of soil or, say, a swab from the human gut, look at it under a microscope, and see billions of thriving, diverse cells. But when they wiped that exact same sample onto an agar plate, maybe, less than 1% of the microbes would actually grow?
Yeah, usually far less than 1%,
Right. So we just assumed the rest were dead or just insignificant.
when in reality, they just hated the menu we were offering them.
Or, they died the literal second oxygen hit them. Or, more commonly, they strictly relied on synthrophic partners, like other specific bacteria, just to survive.
Right, they needed their neighbors.
Exactly. So when culture-independent sequencing came along, specifically whole metagenome shotgun sequencing, it completely democratized discovery. For anyone listening who isn't deep in the bioinformatics weeds, shotgun sequencing basically means we take an entire environmental sample and chemically blend up all the DNA from every organism present into tiny fragments.
Right, you're just smashing everything together.
Right, and then we sequence them all simultaneously and use supercomputers to piece together who is there and what genes they carry.
It is a massive computational dragnet.
It really is, and it allowed us to uncover massive, undiscovered branches of the tree of life. Entire candidate phyla that we had absolutely no idea existed. And most importantly, it allows us to observe these complex networks in situ. We get to see them without ripping them out of their natural context and forcing them onto sterile plastic.
I mean, I don't disagree that sequencing opened our eyes to the sheer volume of microbes out there, but taking a snapshot of fragmented DNA is fundamentally different from studying a functional biological entity.
It gives us the blueprint.
A blueprint isn't a building, though. Without high-throughput cultivation, microbiome science is just trapped in an endless loop of associative datasets. You end up with these massive papers claiming microbiome profile X is associated with disease Y, but you can't prove how and you can't prove why.
But we're identifying their genetic potential. We are seeing exactly what enzymes they carry.
Genetic potential isn't proof. If we want to translate these massive correlation maps into actual medicine, we need living organisms. We have to fulfill modern Koch postulates, meaning we have to isolate the specific microbe, introduce it to a controlled model, and prove that it actually causes the disease or provides the cure.
I hear that.
You simply can't do that with a giant spreadsheet of DNA reads.
I completely understand the demand for physical proof, but to me, moving from the petri dish to the genomic database isn't a loss of biological truth. It's a massive expansion of it.
How so?
Well, Think of culture-independent omics like using satellite imaging to map a dense, unexplored forest. We can see the entire layout of the ecosystem from space. We can map how nutrients flow across the canopy, watch ecological succession over time, and even identify entirely new species just by their thermal signatures, all without ever setting foot on the ground.
The satellite analogy works wonderfully for mapping, but I mean, it completely falls apart when you actually need to understand what you're looking at on the ground. What happens when your satellite detects a massive glowing shape in the forest and you have absolutely no idea what it is or what it does?
Well, you log its presence, and you track its movements across different environments until a pattern emerges.
But what if the pattern is biologically meaningless? You are championing the breadth of discovery, but you are completely ignoring the massive problem of functional dark matter.
Oh, the unknown genes.
Exactly. The recent literature highlights that across diverse microbial systems, 40 to 60% of the genes we sequence cannot be assigned any known biological function. We are talking about 71 million distinct genes sitting in our databases right now doing, well, who knows what.
But we will figure them out over time as our reference libraries grow and improve. It's a process.
But right now, today, it leads to what I call storytelling biopharmacology. Take Do 4295, for example.
Okay, the conserved domain.
Right. That is a highly conserved protein domain, and highly conserved means evolution has protected this specific sequence across millions of years, which usually implies it is incredibly important for survival. It is immensely abundant in the human gut and mouth metagenomes. It is everywhere. And we have absolutely zero idea what its physiological role is. None.
Right, it's uncharacterized.
Exactly. If you're only using a satellite, you're just guessing at what these structures do based on their shape.
Well, I wouldn't call it storytelling. Observing the presence and abundance of these unknown genes allows us to track their ecological significance. Like if a gene like DO4295 suddenly spikes during intestinal inflammation, we know it matters clinically, even if the precise enzymatic function isn't perfectly mapped on a biochemical level yet.
But without the organism, it remains pure speculation. Look at the specific data on aging wild birds from the source material. Researchers used purely observational sequencing data and found that as the birds aged, there was a significant accumulation of transposase genes in their gut microbiomes.
Right. For the listener, transposase are enzymes that allow genetic elements to literally jump around the genome.
Exactly. Jumping genes. They move DNA sequences from one location to another, potentially altering gene expression. So the sequencing data shows a massive increase in jumping genes as the bird gets older. But why?
Well, there are theories.
But does that accumulation represent a passive loss of genomic integrity? Is the bacterial genome just degrading and losing control over time? Or, and this is equally plausible, is it an active, stress-induced transposition mechanism that the bacteria are deliberately using to adapt to a hostile aging host?
Right, the sequence data alone can't definitively answer that specific why.
No, it can't. You need the physical organism to run the metabolic assays. You need to stress the bacteria in a lab and see if it turns those genes on intentionally. You're mapping the shadows of a forest, but you literally don't know if the animals are eating each other or building a nest together.
Okay, but if you're listening to this and wondering why we rely so heavily on these computational shadows, think about it this way. We aren't just looking at isolated genes floating in a void anymore.
Go on.
We have moved far beyond just mapping the ecosystem from afar. We are actually reconstructing the organisms themselves computationally. Genome-resolved metagenomics and the creation of metagenome-assembled genomes, or MAGs, let us reconstruct near-complete bacterial strains.
Right, but reconstruct them computationally, not physically.
Yes, computationally, but the algorithms are incredibly sophisticated. Because shotgun sequencing chops up all the DNA in a sample, we use binning algorithms to look for sequence patterns, like how often certain DNA letters appear together, or how deeply a specific segment is sequenced. We use that to group the fragmented reads back into individual genomes.
Like a puzzle?
Exactly. It's like taking 1,000 different jigsaw puzzles that have been dumped into one giant box, and using the texture and thickness of the cardboard to sort the pieces back into their distinct puzzles. Once we build a MAG,
We can do computational pathway analysis to determine exactly what these microbes are capable of without ever needing to grow them. MAGs are making physical isolates less strictly necessary.
I have to strongly disagree there. Equating a computational reconstruction with a living organism is a massive epistemological error. A MAG is simply not biological reality.
But they represent the genome accurately.
The literature explicitly highlights the compounding, inherent flaws of these reconstructions. Because you are binning shotgun reads from highly mixed, chaotic communities, they are incredibly prone to contamination and incompleteness. Let's look at the accepted standards. A so-called high-quality MAG is considered 95% complete. That sounds robust, right?
I mean, yeah, 95% is very robust genome coverage. It gives you all the core metabolic capabilities of the organism.
The core genome, sure, but empirical analyses show that a MAG at 95% completeness still misses roughly 50% of its variable accessory genes.
Well, accessory genes are just the flexible part of the genome. They aren't the essential, everyday housekeeping genes.
But they are exactly the genes that dictate virulence, antibiotic resistance, and environmental adaptation. If you are missing 50% of the accessory genome, you are missing the exact genetic material that makes that specific bacterial strain dangerous to a human patient.
That is a limitation of current read depth, sure, but the algorithms are improving every single year.
Better algorithms don't solve the fundamental contamination problem, though. Let's talk about the cirrhosis study mentioned in the material. Typical MAG contamination levels below 5% usually pass standard quality control. Most bioinformaticians would accept a 5% contamination rate without blinking.
Right, that's the standard threshold.
But when researchers analyzed it critically, that tiny 5% contamination yielded up to 38% false positive associations in clinical studies.
Because DNA reads from one species accidentally got sorted into the computational bin of another species.
Exactly. Your jigsaw puzzle sorting algorithm failed. So you end up linking a severe disease-causing gene to a specific species of bacteria, and you publish a paper saying, microbe X drives liver cirrhosis. But it turns out that gene belongs to a completely different microbe entirely.
Okay, that's a fair point on contamination.
When you use that contaminated MAG for pan-genome analysis to design a drug, you are targeting the wrong organism. A computational artifact catastrophically distorted the biological reality.
Okay, but if you want to argue that MAGs or computational artifacts aren't pure cultures, laboratory artifacts?
Wait, how is a living, breathing microbe an artifact?
Because a microbe living completely alone in a plastic dish is in a fundamentally unnatural state. When you isolate a microbe on a petri dish, you strip away the cross-feeding. You remove the quorum sensing, which is how bacteria chemically communicate with each other to coordinate group behavior.
Well, yes, we isolate them to study them cleanly.
Right, but you destroy the complex three-dimensional biofilm architectures it naturally relies on. You take this highly social organism and put it in solitary confinement, floating in an artificial, nutrient-rich broth.
It's a highly controlled environment, yes, but it's designed to eliminate confounding variables.
But it's an environment that fundamentally changes how the organism behaves. An isolate in a dish may not faithfully represent its in vivo behavior inside the human gut at all. Gene expression changes dramatically when a microbe isn't stressed by competitors or supported by symbiotes.
That may be true, but
I mean, two bacteria with the exact same genome might express entirely different traits depending on who their neighbors are. You claim cultivation gives you the real organism, but I argue that an isolated culture is just as much of an abstraction as a computational MAG.
it may be in an isolated state, but it is physically responsive. It is metabolically active in real time. And that brings us to the ultimate bottleneck in microbiome science, establishing causality. If we ever want this science to translate into actual therapies for patients, we cannot rely on computational profiles. Take Faecalibacterium prausnitzii.
Ah, yes. A very well-known, highly abundant gut microbe, usually associated with healthy individuals.
Right. Sequencing studies repeatedly associated it with anti-inflammatory states and protection against inflammatory bowel disease. A beautiful correlation. But a correlation doesn't cure Crohn's disease. To prove the mechanism and to actually develop a live biotherapeutic, researchers had to physically isolate it.
Right.
They had to figure out its extreme oxygen sensitivity. They had to physically characterize the specific anti-inflammatory metabolites it produces when it digests fiber.
But my point is, how did they know what specific metabolites to look for in the first place?
Well, let me finish the causality piece because it's critical. To prove it worked, they took those defined microbial consortia and put them into gnatobiotic mice. For the listeners, gnatobiotic mice are raised completely germ-free inside sterile isolator bubbles. They have zero natural microbiome.
Right, a blank slate.
Exactly. By introducing only the physical isolate of fecal bacterium, we can watch exactly what it does to the immune system without any background noise from other bacteria. You simply cannot therapeutically deploy an inferred sequence into a patient's gut. You need the physical bug.
I completely agree that you need the physical bug for a therapeutic, that's undeniable, but you are ignoring how modern culturomics is actually getting its hands on these elusive bugs. You champion culturomics, but traditional culturomics was essentially a blind brute force dragnet.
It was tedious, yes.
You would take hundreds of random petri dishes with different combinations of sugars, salts, and blood and just cross your fingers hoping something grew. It was physiological guesswork. What is saving culturomics right now is sequence-guided culturomics. We are using the very computational mags you just heavily criticized to predict the exact physical growth media required to keep them alive.
Using the genetic blueprint to design the menu.
Precisely. Tools like GapSeq and CarveMe. They extract a genetic repertoire from a computational mag and use complex linear programming algorithms to map out the entire metabolic network of the bacteria. They look for the biochemical gaps.
Right.
Like if the computational genome shows the bacteria lacks the genes to synthesize the amino acid tryptophan, the algorithm flags it. It mathematically identifies that gap and outputs a custom recipe saying, you absolutely must add tryptophan to your petri dish or this organism will never grow.
Which is an incredibly powerful use of the data. I won't deny that.
The recent literature discusses a massive breakthrough using this exact method. Metagenomic data identified that specific, non-obvious additives, things like caffeine or highly glycosylated proteins like mucin, could synergistically enrich targeted, extremely picky gut taxa. They used the genomic shadow to turn the lights on in the laboratory. It proves that a lot of these so-called unculturable microbes weren't actually unculturable.
We just lacked the imagination to formulate the correct media. I don't dispute that at all. In fact, mapping the metagenomic output back to specific media conditions perfectly bridges the two disciplines.
But it goes even further than just tweaking the media recipe. Have you looked closely at the literature on reverse genomics? Because this bypasses physiological guesswork entirely.
You mean the antibody targeting approach?
Yes! Researchers scan these computational mags specifically looking for genes that encode cell surface proteins. They take that DNA sequence, run it through a computer to model the three-dimensional shape of the protein, and find the exposed epitopes, the parts sticking out on the outside of the cell wall. Then, they manufacture fluorescently labeled antibodies designed to bind exclusively to that specific 3D shape.
And then they introduce those antibodies back into the raw environmental sample?
Exactly. You drop those targeted antibodies into a highly complex, messy environmental sample like dental plaque or gut fluid. And out of thousands of different species, only the living target cells light up. Then you use a laser instrument, fluorescence-activated cell sorting, or FACS, to physically pull those ultra-rare glowing microbes out of the crowd one by one.
And the prime example of this, which is genuinely stunning, is the isolation of the candidate phylum Saccharobacteria, or TM7.
Right. TM7 was known only from DNA sequencing for decades. Nobody could grow it. Using reverse genomics, they finally sorted and cultivated them and discovered exactly why they had evaded traditional agar plates for 50 years.
Because they were obligate epibonds.
They were parasites. All three isolated species literally required physical attachment to specific actinobacteria host species just to survive. They physically dock onto the host bacteria to steal nutrients.
Wow.
If you were just using traditional petri dishes, you would never, ever grow them because you didn't know they needed a living host. Without the genomic shadow guiding the way, Culturomics would still be blindly guessing in the dark.
I absolutely acknowledge that the technology linking the computational and the physical is profound, but look at what you just described. It perfectly illustrates my ultimate point. The omic sequence was just a map. The actual physical isolation of the TM7 epibionts is what provided the biological truth.
But you couldn't get the truth without the map.
True, but the map didn't tell you they were epibionts. The MAG didn't explain the parasitism. Only once researchers had the physical organisms interacting in the laboratory could they actually observe the symbiosis under a microscope. The sequencing data generated the hypothesis, but the advanced culturomics validated the mechanism.
So we are definitely arriving at a strong point of convergence here. It's what the field is now calling synthetic ecology or integrated microbiome systems biology. It is becoming clear that neither approach can operate in a vacuum anymore.
They absolutely can't. If you only do omics, microbiome science risks becoming an abstract, almost philosophical discipline of computational correlations that's completely disconnected from biological reality.
But if you only do culturomics without the sequencing data providing unbiased breadth, you are confined to the tiny fraction of microbes that happen to thrive in artificial laboratory conditions. Sequencing identifies the targets, maps the ecosystem from a macro level, and generates the hypotheses that need testing.
Right. While advanced culturomics, aided by omics-driven media design and reverse genomics, physically isolates the living entities so we can validate those mechanisms and actually develop translatable therapeutics. We are finally learning how to use the shadows to catch the entities that cast them.
And this tension between computational ecology and experimental microbiology is exactly what pushes the field forward. It forces bioinformaticians to confront the physical realities of living organisms, and it forces traditional microbiologists to look far beyond the edges of their agar plates.
There is an incredible amount of depth to explore in the recent literature regarding these methodologies, especially as we start applying them to complex clinical diseases.
Thank you for joining us on this exploration. We'll leave you to ponder that original image. Whether you prefer scanning the vast dark forest from a satellite or tracking the living, breathing animal on the ground, uncovering the true shape of the microbial universe requires us to do both.